Hello, welcome to another screencast where this time we'll be discussing hemostasis. That is the body's homeostatic ability to keep a steady state of the blood. That is to stop hemorrhaging, right, or stop bleeding here. So what actually happens there? So you should be able to already describe and sort of explain the need for hemostasis, why the body would need to do this. But then how is it done? So we're going to focus on the mechanisms of hemostasis. So the majority of the screencast is going to deal with that. And then finally, we'll end by talking about some conditions or pathologies associated with abnormal clotting, either lack of clotting, like in a hemophilia, which you've probably heard of, maybe not heard of deep vein thromboses, where this is going to be too much clotting. You might have heard of embolisms. We'll talk about a pulmonary embolism as an example. And then another one you probably have not heard of is thrombocytopenia here, conditions associated with clotting abnormalities. So just want to start this off by saying that there's four basic things that are sort of going to be happening here. Right? The first thing, that anytime that you have any sort of blood loss, is you're going to first have vascular spasms. So vascular spasms, meaning vasoconstriction. So the blood vessels are going to constrict, right? Namely arteries are going to constrict here. The second thing that is going to happen is going to be a platelet plug formation. So you're going to form this thing called a platelet plug. That platelet plug is going to be overlapped and followed and overlapped by coagulation. And then the fourth aspect here is fibrous tissue forming. Right? And then the sort of fifth one that follows from that is fibrinolysis. So this is actually, you can kind of think of this as like 4A and sort of 4B instead of 5 different things, because these things are pretty closely linked together here. So vascular spasms, platelet plug, coagulation, fibrous tissue followed by fibrinolysis. So this is happening when you rupture a blood vessel, right? When blood vessels break for some reason. Okay, so this diagram here is going to be is basically talking about the platelet plug formation, the coagulation, and then the fibrin formation. Right? So the stimulus for vasoconstriction, now let me just go back a second and just address this. So the stimulus for this to happen is any sort of direct injury to the blood vessel, and any sort of, and also pain receptors in the blood vessels release chemicals that will signal the smooth muscle to contract. Signal the smooth muscle, right, there to contract and squeeze off blood flow. So this should be obvious, right, that if you're going to vasoconstrict upwards of the damage, you're not going to be losing any more blood, right? It's limiting blood flow to that area. So that's the first thing that's going to happen, right? But surely following that is you're going to have this platelet plug formation. Now, I highly recommend running the PowerPoint on your own and then clicking on these animations and watching them. Although I think that 1 and 2 here are the same thing. But both of that's good, and then also animation 3 is very useful as well. But here's the platelet plug formation. What's going on? Let me talk at you a little bit about anatomy and remind us the anatomy of a blood vessel. This light pink stuff here is the basement membrane, right, or areolar connective tissue, right? And remember that this loose connective tissue contains fibers of collagen as well as elastin, but it's going to be collagen that is most important here. That basement membrane is then in contact with these reddish pink cells here that are the endothelial cells. And those endothelial cells are smooth. They line the blood vessel there. Right? So in the blood vessel then you see this white area, and that white area is the plasma. Right? These are red blood cells, right? And then the green things here are platelets or thrombocytes. Platelets, thrombocytes. Okay, so we're talking here about platelet plug formation. What's actually happening is that when you rupture that blood vessel, right, seen here, right, you damage the endothelial cells. Those endothelial cells break, and when those endothelial cells break, they send out chemical signals. When the collagen fibers are exposed, that is going to send out a signal, right, to the platelets. So these collagen fibers loose here, right, will also stimulate then platelets to come to the area. So what's going to happen, I want to detail this on a different slide, this from a different textbook that's not as much detail as your textbook does. So let's take a look at this diagram for just a second. So this is a cross-section of the blood vessel here, and we see that this is the damaged area, right, where here's the smooth muscle that's been damaged, here's the collagen fibers in the basement membrane, right, and then here are your endothelial cells, right, lining that, so it's a simple squamous epithelium there. So they've all been damaged. So I've got collagen exposed here, I got damaged endothelial cells here, and this damage, this collagen, and the damage to these endothelial cells, there is something called von Willebrand factor, or VWF. The von Willebrand factor are these little black dots here and here and here, right? And here and here and here. This von Willebrand factor will signal platelets. Here's a platelet here. It will signal the platelets to come and bind to them. So the collagen and the endothelial cells have this von Willebrand factor that they begin producing when they are damaged. The von Willebrand factor then causes the platelets to bind there. So not only that, you have the platelet binding, but then as soon as that platelet binds, that platelet begins to release other chemicals, ADP, adenosine diphosphate, this stuff called thromboxane here. And these are chemical signals then to tell other platelets to actually change their shape. So the platelets start changing shape. They form these star-shaped appendages here, right? And then they start producing this substance, this protein called fibrinogen, right? And they produce these fibrinogen receptors, and essentially this fibrinogen with these fibrinogen receptors helps to stick one platelet to another. So you see here, here's an appendage, an appendage, an appendage. This platelet, right, and these platelets here are able to sort of cross-link with platelets that are bound here. And this is what's going on with the platelet plug. This is what we mean by a platelet plug formation, right? So you should pause here and take a second and sort of down in this area here, make yourself a little flow chart and diagram, right? What happens first, second, third, the von Willebrand factor and the changing the shape and the binding here to get the platelet plug formation. We can go back now and look at this diagram and see what's happening here. So here's our platelet plug being formed, right? And not only are those platelets, when they bind to each other, forming the plug, they will also then be signaled to release serotonin, right, which causes other vessels to constrict. Right? Vascular spasms occur. The platelets also release chemicals that attract more platelets to the site. Right? So when you go back to here, right, not only do you get all of this platelet plug stuff happening here, right, but this thromboxane and this ADP, right, will signal vasoconstriction and will also call other platelets to the area. Okay, so lots of stuff going on right there, and exactly the type of stuff that we could ask on a timed writing. So after the platelet plug formation, after platelet plug formation is going to be coagulation. Now this diagram is extremely complex, and I don't mean to have you memorize any of this. But what I want to point out is that right here we've got tissue damage, and that is, again, we have the endothelial cells rupturing here. This is what we mean by tissue damage, right? And the cells external to that, so smooth muscle cells, endothelial cells are damaged, and then also contact with collagen here of the damaged blood vessel. So both of these, this extrinsic pathway and this intrinsic pathway, ultimately, bottom line is that they lead to the formation of this fibrin and a fibrin clot. So this is what coagulation is, right? Now, in between this damage up here and the fibrin clot forming down here, you notice that you have lots and lots of steps here, right? Thromboplastin, here's factor 7, right? Factor 12, 11, 9, 10, all of these factors right down here. Here's factor 13, etc. Right? So all of these factors, right, these are all clotting factors. You need all of these in order to get this fibrin clot to be forming. Some of these factors here are relying on calcium ions, right? Here, vitamin K, not shown on here, but vitamin K is also involved with this formation here, right, of this fibrin clot. So let's go back to that original diagram up here and see what's happening in the coagulation events right here. So this tissue factor, right, released, right, by the damaged cells, this would be the extrinsic pathway, and then the platelets and the tissue factor, calcium, vitamin K, other clotting factors, this is related to the intrinsic pathway there. Right? But what's happening, bottom line, is that those clotting factors are going to activate this prothrombin stuff. So all the stuff in the green box here activates this prothrombin activator. Prothrombin activator is an enzyme. Okay? This enzyme will turn prothrombin into thrombin. Right? So prothrombin is an inactive enzyme. Thrombin is another enzyme that is now active. And this thrombin enzyme will turn fibrinogen, which is soluble, into fibrin. So this thrombin, again, turns fibrinogen into fibrin, which is insoluble. This fibrinogen is a protein that is dissolved in the plasma. So if you go back a couple weeks and look at the contents of blood, you remember that plasma, there's a lot of dissolved proteins in the plasma there. So the protein dissolved in the plasma, it becomes this insoluble now fibrin. And if you follow the yellow arrow up, this fibrin is going to now infiltrate this platelet plug. And so this fibrin is a protein that gets laid down in that platelet plug and begins to trap red blood cells, white blood cells in there, some thrombocytes as well. And then this fibrin protein traps cells, right, etc. And this is what is forming a clot. Right? This is what the coagulation is. Eventually then, that fibrin will contract. This protein actually shorten or contract and pull the damaged area together. And so here you see the damaged area is not together. Here you see that it is. And pull the damaged area together, right? And you get new cells growing in there. You get new collagen being laid down by fibroblasts. You get new endothelial cells being laid down in there. And this then repairs it. So all this timing is just about right. So again, this is the coagulation part of that here. I'm going to stop here and I'll do the pathologies in a separate screencast. Right? So the basic mechanisms of hemostasis, again, I would highly recommend stopping this right now, going to the PowerPoint and looking at, again, these animations, especially 1 and 3. 3 is kind of funny. Right? The guy's voice is really weird in that one, but the animation ends up being pretty good once you get over his his silly voice, all right? So let me stop there, and if there are questions, jot them down, talk about them with each other, and then when I come back to class, we'll discuss it.